Deep in the Altai Mountains of Siberia, where permafrost preserves secrets from the Pleistocene, a single fragment of a pinky bone lay undisturbed for over 40,000 years. This unremarkable fossil, no larger than a pebble, would become the Rosetta Stone of human evolution, containing DNA from a previously unknown human species. The field of paleogenomics has transformed these molecular time capsules into revolutionary insights, allowing us to reconstruct entire genomes from mere specks of bone powder. Through staggering advances in DNA extraction and sequencing, scientists can now read genetic code older than civilization itself, rewriting our understanding of human origins with each new discovery. This isn't just science. It's a forensic investigation spanning millennia. The story begins with a scientific heretic, Svante Pääbo, a young researcher who defied conventional wisdom in the 1980s by attempting the impossible, extracting DNA from Egyptian mummies. At the time, experts believed no genetic material could survive beyond a few thousand years, yet Pääbo's breakthrough proved them wrong. The challenges were Herculean. Ancient DNA is typically fragmented into pieces shorter than 50 base pairs, degraded by millennia of environmental exposure. But Pääbo's team at the Max Planck Institute developed techniques to amplify and sequence the genetic codex establishing paleogenomics at the forefront of the human origin story. Paleogenomics is the recovery and analysis of ancient genomes from organisms long extinct. Unlike traditional paleontology, which studies fossils and bones, this field deciphers the molecular genomic information, providing insight into evolutionary history, past population migrations, and the genetic makeup of extinct species. The process is laborious, beginning with ultrasonic cleaning of fossils to remove surface contaminants, followed by drilling into dense bone regions least affected by decay. Extracted DNA is then treated with enzymes that repair some damage, while next-generation sequencing reads the shattered genetic code millions of times over. Advanced algorithms stitch these fragments together, like assembling a prehistoric jigsaw puzzle where 99% of the pieces are missing. The result? A window into evolutionary biology so precise it reveals not just what species existed, but how they lived, adapted and interacted. Perhaps even more revolutionary is paleoepigenetics, which was born from paleogenomics. Paleoepigenetics is the study of chemical modifications that regulate gene expression in ancient DNA. While paleogenomics focuses on the genome, the hardware of human biology, paleoepigenetics focuses on the epigenome, the software determining which genes activate under specific chemical conditions. Scientists can detect DNA methylation patterns in fossils, revealing how environmental pressures shape gene activity in ancient humans. For example, epigenetic analysis shows that Neanderthals had distinct methylation in genes related to limb development, possibly contributing to their robust skeletal structure. Similarly, Denisovan epigenetic profiles suggest adaptations to hypoxia, aligning with their high-altitude habitats. This field doesn't just tell us what genes archaic humans had, it reveals how those genes behaved. This gene expression offers unprecedented insight into their physiology and even behavior. In 2010, after 4 years of painstaking work, Pääbo's team published the first complete Neanderthal genome, sequenced from 3 individuals from Vindija Cave, Croatia. The findings sent shockwaves through anthropology. Modern humans outside Africa carried 1 to 4% Neanderthal DNA, proving interbreeding occurred around 55,000 years ago. Even more striking, Neanderthal DNA influences modern immune systems, with their HLA gene variants appearing in up to 50% of Europeans. This wasn't just ancient history, it was living biology, with Neanderthal genes still active in people today. But perhaps the biggest revelation from Parbo's work focused on the analysis of a 41,000-year-old finger bone from Denisova Cave. Researchers had expected to find Neanderthal DNA. Instead, the genome revealed an entirely new human lineage, the Denisovans. This species, unknown from fossil records, was identified purely through paleogenomics. Comparisons showed that the Denisovans diverged from Neanderthals around 400,000 years ago and interbred with modern humans in Asia. Today, indigenous Australians and Papuans carry up to 5% Denisovan DNA, while Tibetans carry the Denisovan version of the EPAS1 gene. The discovery fundamentally altered the human family tree, proving Homo sapiens at one time shared the Earth with at least two other intelligent hominins, the Neanderthals and the Denisovans. We now know that modern humans interbred with archaic species multiple times across different regions. Some populations, like Melanesians, carry genetic contributions from both Neanderthals and Denisovans. The archaic DNA wasn't just passively inherited either. Natural selection actively favoured certain genes, like those involved in immune function and metabolism. This complex legacy means that, genetically speaking, there's no such thing as a pure modern human. We're all hybrids of multiple ancestral lineages. This archaic DNA continues to impact modern humans. This genetic legacy has resulted in adaptive introgression attributes whereby a gene from an extinct species provides a critical advantage to modern humans. The Denisovan-derived EPAS1 gene variant is carried by modern-day Tibetans. This gene variant enables Tibetans to experience 40% more oxygen saturation than normal human beings at high altitudes. The gene prevents excessive red blood cell productions, reducing the risk of high-altitude sickness and hypoxia. Similarly, Neanderthal DNA has shaped the immune systems of some modern-day European. Many European inherited the Neanderthal version of toll-like receptor, TLR, genes, which plays a crucial role in pathogen detection and immune system responses. However, this genetic legacy is a double-edged sword. The same Neanderthal DNA that once provided protection now correlates with increased risk for autoimmune disorders like lupus and Crohn's disease. Even more strikingly, during the recent COVID-19 pandemic, researchers found that a Neanderthal haplotype on chromosome 3 actually increased severe disease risk. However, contrary to this effect, the Neanderthal haplotype on chromosome 12, OAS1, 2 and 3, actually had protective effects against severe COVID, a profound impact. Beyond immunity, Neanderthal DNA affects modern human traits ranging from hair texture to mood disorders. People with certain Neanderthal variants report higher pain sensitivity, likely an adaptation to colder Pleistocene environments. Other archaic genes influence skin pigmentation, metabolism, and even nicotine addiction. A bizarre legacy considering tobacco didn't exist in Eurasia until recent centuries. Perhaps most intriguing are findings linking Neanderthal DNA to brain development, with some variants associated with altered neuronal connectivity. This raises profound questions about how archaic admixture shaped cognition and behavior in modern humans. It seems we are only beginning to scratch the surface. The ancestral genetic legacy is fascinating in its complexity. Beyond the EPAS1 gene, ancestral humans contributed variants affecting fat metabolism and blood sugar regulation adaptations, possibly linked to their high-altitude lifestyle. Even more remarkably, some Southeast Asian populations carry archaic DNA sequences that appear to improve olfactory receptors, potentially enhancing smell sensitivity. Meanwhile, in the Arctic, Inuit populations show evidence of positive selection on ancestral genes involved in fat storage, crucial for surviving extreme cold. These paleogenomic findings demonstrate how archaic DNA provided positively tailored solutions to environmental challenges facing our ancestors. This natural selection has found its way into modern humans facing similar challenges. This research has profound implications for modern medicine. Many diseases, including diabetes, depression, and autoimmune disorders, show links to archaic DNA. Pharmaceutical companies are now investigating how Neanderthal-derived biological pathways might inform new treatments. Conversely, some archaic variants may be beneficial. Researchers are studying whether Neanderthal immune genes could help combat antibiotic-resistant infections. This merging of paleogenomics and medicine represents an entirely new approach to human health, one informed by deep evolutionary history. The field continues to accelerate with new technological breakthroughs. Single-cell DNA sequencing now allows analysis of individual ancient cells, while proteomics can extract information from fossilized proteins when DNA is unavailable. Most excitingly, scientists are attempting to synthesize Neanderthal genes and insert them into human stem cells, allowing direct study of their biological effects. These advances promise to reveal even more about our archaic inheritance and what makes modern humans unique. Svante Pääbo's 2022 Nobel Prize recognized not just his individual achievements, but the birth of an entirely new scientific discipline. Paleogenomics has fundamentally altered our understanding of human identity, proving that Homo sapiens are not a singular lineage, but a tapestry woven from multiple human species. This paradigm shift echoes beyond science, challenging long-held notions about human uniqueness and our place in nature. Every time a Tibetan breathes easily at high altitude or a European's immune system fights infection, one truth comes clear: the genomes of our archaic ancestors live on within us. The following documentary is titled Paleogenomics: Unlocking the Hominin Genome, produced by Sclera Documentary Films and written by Dr. Meralli.